I work now on a fungus that some of you work at here at BBI, but I think from another point of view. And I think along with what I've done in the past, I always look from the fungus perspective. So I try to understand the cells of the fungi that grow vigorously and why they grow as they grow. And I'm not so worried about the fungus degrading plant material, which is your main interest here. So I'll try to put it in a perspective so that it's of interest as you as well. And we've been focusing on the mechanisms or the enzymes that participate in the cell wall growth of the fungus. So some of you are familiar with this fungus, Neurospora crassa, because several of the PIs in this institute work with it. And I'm going to try to use this. I cannot see the cursor here. I was trying to use that because somebody else is looking at it, but I have to use the pointer, sorry. So this is Neurospora crassa covering a petri dish with conidia, with the asexual spores. And if we look closer under the microscope, this would be one of the cells of this fungus, a hyphae with the branches. And here you were seeing something that moves very fast. This is the tip of a hyphae. And at the very tip, there is this structure, the Spitzenkörper, which means apical body in German. And we've been mainly interested in the functioning and the organization of this structure, which was kind of mysterious for many years, the composition. But it was clear from very early ages in fungal biology that it was a determinant of the growth of the fungus. And if this structure disappears, the fungus arrests. Therefore, intuitively, we knew that there was something about this structure important for maintaining the growth of the fungus. And that's what we've been working on finding out what is composed of and how it works. And if we look at this, it's a three-dimensional reconstruction from EM CICESE section done by Girbar in 1969. We can see this accumulation of vesicles here at the very tip, in which we've seen before that the Spitzenkörper is located in the hyphal tips. And as I mentioned before, we believe that it has a major role in apical growth and hyphal morphogenesis. So over the years, we have been identifying the components of this structure, and we have identified vesicles primarily of both types, and we'll go into detail of that today on micro and microvesicles, also actin microfilaments, ribosomes. There are a lot of ribosomes inside this structure, and we still don't understand what they're doing there, but that's another area of research. And then an amorphous matrix, which we have no clue of what it's made of. And around 1989, this model was proposed. It was a model to understand how hyphal morphogenesis is determined. And this model proposed that this Spitzenkörper behaves as a vesicle supply center that collects vesicles that are synthesized in other parts of the hyphae. The model didn't consider the source of these vesicles, but this vesicle supply center accumulates temporarily these vesicles here at the Spitzenkörper, and then they are released in all directions towards the plasma membrane, where the membrane of the vesicle fuses with the membrane of the cell and expands both membrane and contributes with new proteins to make more cell walls. So it's the movement of this vesicle supply center, the advancement of this vesicle supply center as it releases vesicles in all directions, what determines this hyphal shape. And then this shape was determined by a very, very simple equation that is below here. So our questions were focused on really identifying the biological components that could prove this model right, and to identify the source of vesicles and the fate of these vesicles. Now, let's look at some facts. I don't have to convince you why fungi are important, but link it to where I was saying before. I think some of the main characteristics important for us in the filamentous fungi is that they have a much higher capacity for protein secretion than yeast. In the case of Aspergillus and Trichoderma, which are other fungi that are model systems in fungal biology, the strains can yield up to 100 grams per liter. And also a very important fact is that they can grow apically. This tip growth that I've shown you can go on and on under the appropriate substrate forever. So they can present apical growth indefinitely, unlike yeast cells that only polarize when they bud or when they mate in the mating tubes, or in pollen cells that also show apical growth, but they have a very determined length of polar growth, or neurons, that is the same situation. So filamentous fungi are excellent examples to study apical growth because they can continuously grow apically and secrete if they have the right conditions. In biotechnology, there has been a search for these fungal mutants that branch a lot, with the idea that if they produce a lot of branches, then they have more tips, and since secretion has usually been assigned to the hyphal tip, then you would have cells that hyper-secrete. However, that's not the whole story, because most of the cases, mutants that make a lot of branches, they're affected at some very important steps in cell growth. So even if there was at that time a search for those mutants that hyperbranch, I think that's not the whole solution to the problem. So it's very important to study, for all of what I said, apical growth and how this apical growth is generated. And our goals in the lab have been to elucidate how the hypermorphogenesis is achieved, to identifying these vesicular components of the Spitzenkorper, and then study the traffic and secretory pathway. I took here pathways because every time you discover one, then it turns out that there is another one. The things get complicated and more complicated. But basically, the pathways that are associated with these vesicles that conform the Spitzenkorper. So if we look at the cell wall, just a very fast review of the main components of the cell wall of the fungi. There are some structural components. The main one is chitin and chitosan. Is a deacetylated form of chitin that some fungal forms present, and beta-1,3,1,6-glucans. And then a matrix component, the amorphous components of the wall that are glycoproteins and alpha-1,3-glucans. And we have started by looking at the enzymes that make chitin and glucans, the structural components that give rigidity to the cell wall. And this cartoon represents how we think that these enzymes that make the wall are assembled and get to the plasma membrane, because these enzymes they have transmembrane domains, and we think that they are transported in vesicles, in different types of vesicles, and I'll show you now why we think that's true. And these transmembrane enzymes get incorporated from the vesicles into the plasma membrane, and they synthesize these fibrils of chitin or glucans outside the plasma membrane into the cell wall. So they get the substrates from the cytosol, and they incorporate them in the growing cell wall. So in terms of chitin, the family of chitin synthase is quite large. In filamentous fungi, there are about 7 classes of chitin synthesis. In yeast, there are only 3. So filamentous fungi have homologs of the 3 classes. These would be the ones in yeast. But then they have additional chitin synthases. Some interesting ones are these ones that have a myosin motor domain in the amino terminus, and then there is this chitin synthase 6, which is quite different to all the others, and we don't know quite yet what is the function of this class, but in some plant pathogens it's been shown to be important for pathogenesis. So what we did is to start with GFP each of these chitin syntheses, and we found this is an example of chitin synthase 1. I don't know if you can see okay, or we should turn down. Oh yes.
What's the role of cytochrome B5 in the chitin synthase?
On this one? So in one of the families, right?
3 of them.
Yeah, this one here. So I don't know what's exactly the role of that. The catalytic one is the one that has been well studied, that is important for the synthesis. But yeah, in this case, we don't know exactly what the role is. So for chitin synthase 1, we found accumulation of these enzyme here at the Spitzenkörper. You can see at the tips of branches and apex. In some cases, you can see it also at the plasma membrane. I told you that this is a plasma membrane protein, and in a lot of cases you don't see it, but we think this is because the Spitzenkörper is really masking, the area of we're looking at the central point here, the focal plane of the center of the cell. So when you go a bit off, then you can see it in the plasma membrane. And then you can see it as well in the forming septa. So you can see here a 3D animation, and you can see septa that have been formed, and they leave a pore in the center. And in this case, you'll see this is an overlap of phase contrast microscopy and fluorescence microscopy, and you see the Spitzenkörper here advancing. When we look at the remaining chitin synthases, we saw that all of them are found here at the core of the Spitzenkörper. This is a staining with FM464 that we use as marker of the Spitzenkörper. But as you see in the colocalization, there is only yellow at the very center. So the chitin synthase enzymes are localized at the center of this Spitzenkörper. And when we looked at glucan synthase, surprisingly, we saw that glucan synthase was found at the outer layer of the Spitzenkörper. So in this cartoon, you see chitin synthase, in this case tagged with M-cherry, at the center, and in the outside, you have the vesicles that contain glucan synthase. We did cell fractionation of these cells to analyze the activity and to see if we could actually see them associated with different populations of vesicles. And you can see here in the bottom a Western against GFP of the chitin synthase 6 GFP tag strain. And you can see the most intensity of this band coinciding with the peak of chitin synthase activity. And in this case here, this is the Western for the strain where GS1 has been tagged with GFP. And you can see that the highest intensity of the bands coincide with the two main peaks of glucan synthase activity. So it seems that there are different populations, density-wise, of vesicles with different cell wall building activity and different organization in the cell. And if we look at the ultrastructural level at the tips of Neurospora, you can see this would be the area that correlates with the Spitzenkörper in vivo. And here at the center where the asterisk is masking what I want to point, here there are microvesicles. These microvesicles are about 25 to 40 nanometers in diameter. And in the outside here, or here, this is a zoom, you can see the macrovesicles that have range between 70 to 100 nanometers in diameter. So I said that all chitin synthase coincide in the core of the Spitzenkörper. So this is a colocalization of 5 and 1. And if you look at the merge image, you see that everything is yellow, or the same thing here. But if you do a zoom at this image, you can actually see that they are not the same vesicles. That, yes, they colocalize at this area, so they converge at the core of the Spitzenkörper, but they seem to be traveling in different populations of microvesicles or chitosomes, which are these microvesicles that carry chitin synthase. And if we look at septa, the same could be seen. If we look at this image here, it seems that chitin synthase 1 and 5 colocalize perfectly in this form septum. But if you look at reconstruction in a transversal cut, then you can see that they don't fully colocalize. And if you look at this animation, which this is where this is taken from, you can see here clearly that they don't fully colocalize. So all the evidence suggests that there are different populations, subpopulations of these small vesicles that carry different types of chitin synthase. And this would be the view of how septum is formed. We don't see the traffic of vesicles, and I think I'll show you later why. I think it's just a very fast mechanism of traffic that we cannot resolve at the resolution of this microscopy. But we believe there are vesicles that reach the areas where the septum is being formed, and the fusion of these vesicles with this growing plasma membrane is the mechanism that allows new plasma membrane to be formed at septa sites. And some late studies have shown, in fact, that some secretory vesicles or some secreted enzymes are also directed to septa, not just to tips. So this general idea that we had some years ago that secretion occurs mainly at tips of the fungal cells is not quite true. There is a lot of secretion, it seems like, that occurs at different areas, at septa and subapical regions of the cells. And I think we still don't understand why secretion is directed to all these areas when growth of the fungus is mainly directed to the tips. So maybe there is some division of tasks in the fungal cells. So our other interest is to find out where these cytosomes, these microvesicles that contain chitin synthase, are generated. And this came from some evidence we got some years ago that when we use brifellin A, which is a drug that is used traditionally to interrupt ER2 processes because it interferes with SEX7, the gap for this alpha protein, which participates in early Golgi events, we saw that chitin synthase 1 was not mislocalized from the Spitzenkofer. And then we thought that perhaps it was not using the traditional ER2 Golgi pathway. But in yeast, and this comes from the work in the Sheckman lab, another, and Trevor also has work in chitin synthase 3 in the yeast cells in Saccharomyces. So they have found that chitin synthase 3 in Saccharomyces goes from the ER2 Golgi pathway. They need the exomer, coat protein, that participate in these late steps of the secretory pathway to, so it's like a sorting mechanism to decide where to go into the plasma membrane. And then once in the plasma membrane, they need also the action from some activators for this enzyme to be active and start making chitin synthase. However, they also found that there are some mechanisms that there are alternative pathways independent of the exomer and that they are controlled by some adaptor protein dependent signals. So it seems that the fate of this chitin synthase is quite complicated and is determined by different domains in the amino acid sequence of the protein. So it's quite a complicated mechanism, and we have 7 different enzymes to study. So we're just starting now to study what's happening in Neurospora. Neurospora has homologs of all these proteins identified in yeast for the traffic of chitin synthase 3. It has homologs of the ER export chaperone. It has only one homolog of all these chaps that constitute the exomer, and it has 3 homologs of chitin synthase 4, of the ones that activate the enzyme at the plasma membrane. And perhaps this is a reflection of the different types of chitin synthase that we find in Neurospora, and each of them could be activating a different division of chitin synthase. This is just wild hypothesis. We are really starting to analyze that. But what we did, having these GFP tags, is to precipitate the proteins, the putative proteins that interact with this chitin synthase, and we did that for chitin synthase 1, 4, and 5, and we found YTT1, which is a RAB GTPase that I'll talk about it more later on. Just remember this one, YTT1. We found SEX14 that is also involved in the secretory pathway. And I only listed here the ones that are shared by the 3 or by 2. And on gray are some that they are not really well supported with these 2 rounds of analysis, but they showed up, and I think we just need to do it once more to see if they are really interacting with them. We didn't find any of the exomer proteins that I showed you before, but that may be due because they are not very strong interactions, and that's why we didn't find them in this pull-down acids. But we found some interesting ones that we're going to look at. They are the serine protease and a protease inhibitor and this phosphatase. And I think that's interesting because in the past, some of these chitin synthases have been reported to be zymogenic. They need to be proteolytically activated, but they've never found the activators of these chitin syntheses. So I think at least we have something that we can now test and see if these are at all involved in activating the at least chitin synthase 4 and 5. And as you see here, there is a septin, which is involved in it's part of the cytoskeleton, and that perhaps has something to do with this myosin motor domain of chitin synthase 5. So right now, we're just starting to really look at these interacting proteins and do protein-protein interaction assays to test it. So for the second part of the talk, I want to focus on the traffic of these vesicles. And as you probably heard, being here in this campus, vesicle traffic is highly regulated, and it involves a lot of steps and coats and tethers. So vesicles are first budded from a donor membrane. They are covered by several coats that are very specific depending on the cargo. They are then transported. They are tethered or getting close to the acceptor membrane with the help of tethers and RAP GTPases, and then the final step involves the fusion, which also is a very specific process where the bis net of the vesicle interacts with a cis net of the target membrane until both membranes are fused. And we've been looking at some of these components, the tether and the RAP GTPase. So first we look at the exocyst components in Neurospora. Neurospora has one homolog of each of the 8 components of the exocyst, and it's an octameric tether that participates in these last steps of the secretory pathway, as I was mentioning. And what we found is that some of these components here, Sec5, 6, 8, and 15, are localized at the very apical part of the plasma membrane of the hypha, and 3 components, Sec3, Exo70, and Exo84, are closer to the macrovesicular area of the Spitzenkorper. So even if biochemically we couldn't test that there are 2 subcomplexes, we think that, well, Sec3 is actually like in the middle, it's not so clear, but we think that there are 2 different organizations, that some subcomplexes are attached to the macrovesicles and some others are in the plasma membrane, and it's the association of both subcomplexes that forms the whole exocyst complex that helps the fusion of the vesicles with the plasma membrane. If we look at one of these components, Sec6, by search microscopy, search microscopy allows to image the very external part of the cell close to the cover slip. So in this case, we're imaging about 200 nanometers of the periphery of the cell. And you can see here how it's not, there are pulses almost of exocytosis. It's not a continuous fluorescence, a crescent that, as I was showing you in the previous slide, you can actually see points of fluorescence, in this case it's in black and white. Can you see here? So this is from this movie, and you can see how the fluorescence keeps going from one end to the other, from one side to the other. And we think this is a reflection of these vesicles being exocytosed at a given time. And here you can see how this represents each peak of fluorescence reaching the apical membrane. So you can see how this keeps alternating. Yeah. So when we did for expression of these exocyst subunits and what we had before, chitin synthase 1 or glucan synthase, you can see that there is no colocalization of this Spitzenkorper and Sec6 or chitin synthase 1 and exo 70 surrounding the Spitzenkorper. But when we did colocalization of exo 70 with GS1, they are both at this external layer of the Spitzenkorper, and here again there is no colocalization. And you can see that better here in this movie of SEC6 and chitin synthase 1, in which maybe you can only see sometimes a bit of yellow of one of the vesicles that is reaching the apical bit of the cell. Now, in terms of mutants, only SEC5 was a viable or semi-viable mutant for Neurospora. It grows very bad. This is a deletion mutant made by the Genome Project of Neurospora, and this is a UV mutant. And they are both highly affected. They have a very slow growth. Neurospora covers the plate in about 1 to 2 days, and this is after a week or so of growth, and they just grow very compact, highly branched, and as you see here, with hyphae very distorted and no Spitzenkorper. This is a staining with FM464. So when we looked at the electron microscopy, this is an EM image of a wild-type tip of Neurospora. That's the one I showed you before as control. And if you look here at the tip, we caught an exocytic event in which some vesicles are fusing with the plasma membrane. This is a very rare event. It's kind of hard to capture the cell in that state because exocytosis is very quick, so you have to really, even if these are cryophic cells, it's not very often that you see these fusions. But this is SEC5 mutant, and as you can see, the cell is just full with vesicles, but full with these big vesicles. You still see a core here. This organization has not been lost. And at this point, we don't understand still why, but at least there is a little core of microvesicles. But the cell, it seems that with no exocyst, cannot undergo the exocytosis of big vesicles, and they accumulate in the cell, similar to what has been reported for the yeast mutant. And what we have been doing more recently is to look at some of the RAB-GTPases that I mentioned before participate in this regulation of the vesicular traffic. And RAB-GTPases alternate between a GDP-bound inactive state and a GTP-bound active state. And we focused first on the homologue of the mammalian RAB1, YPT1 in yeast, and we follow the same nomenclature than in yeast. So I'll be talking about GPT, even if they are not yeast proteins, but they are the homologues of the yeast protein. And YPT1 is one of the RAB-GTPases that participates in several steps of the secretory pathway. It participates in Golgi events, but it also participates in traffic from early endosomes to Golgi. And more recently, it has been found that they also participate in the formation of the autophagosome. And as I mentioned, also we've looked at SEC4 and YPT31, which are both post-Golgi RAB-GTPases, so they are supposed to participate in late steps of the secretory pathway. So when we look at YPT1, we found this panca all along the cells with discrete points of fluorescence, but then we saw also major accumulation of fluorescence at the tip where the Spitzenkorper is. And that was kind of shocking. We were not expecting that. Then we went on and we tagged YPT31, this post-Golgi RAB, and we saw that it accumulates at the external layer of the Spitzenkorper. And with step 4, which was harder to get, this one, it was hard to get defined label. It's more dispersed. In some cases, in other systems, they use a triple GFP, and perhaps that's what we have to do to see the fluorescence clearly. But you can still see that it's accumulated basically in the surrounding area of the Spitzenkorper on the outer layer. And finally, I'll show you this SIN1, which is a snare that is localized. It seems in all the region of the Spitzenkorper. So we were not expecting this so much because this was done in parallel with the chitin synthase work. It was not done after we got the other results. But it seems that there are different types of RAB-GTPases that associate with different types of vesicles in the Spitzenkorper. And that's very nice because now it's sort of proved what I showed you before, that YPT1 was precipitated with all the chitin synthase in the mass spec analysis. And in this slide, you can see co-expression analysis of chitin synthase 1 and YPT1. This is failing here. YPT1 in the core and chitin synthase 1 in the core, and here they co-localize. But when we co-express YPT1 and YPT31, you see that one is found in the core and the other in the surroundings. Here, some of the fluorescence is washed out because in this co-expression, you don't get sometimes the same amount of fluorescence for both fluorophores. But I also mentioned that YPT1 is found in this puncta along the cell, and we thought that these were putative Golgi cisternae. So to show that, or to confirm that, we also labeled some other markers of Golgi compartments. And we started by labeling US01, which is a Golgi tether. It's a long coil tether that has been found in early Golgi cisternae. And yes, they were found in also some cisternae, but we had to do some analysis to really show that they colocalize. So here you have the YPT1 channel, US01, and here the merge. And some of these arrowheads, they show the cisternae that colocalize. So when my student did the correlation quotient analysis of this, he found that they were 0.14. The correlation index was of 0.14, which is, according to the standards, is a good level of colocalization or partial colocalization. If the values drop below 0, up to -0.5, that means that there is no correlation, there is an exclusion. And in this animation, you can see the messy Golgi cisternae moving along. Generally, there is an anterograde movement of the Golgi cisternae, and you can see some cases here that these points colocalize. Oh, and incidentally, I mean, this is done in the magenta and cyan, just in case that there is some colorblind person in the audience. We never thought about it until somebody pointed it out. We were showing always in green and GFP and red for cherry. So that's why we have this feminine color, even if the author of this is a man. So we also use this marker to look at the late Golgi. This is Golgi-associated retrograde protein complex component. We tagged BPS 52, and we saw that there was also some partial colocalization with YPT1 with values of 0.18. So we think that some population of YPT1 colocalizes with early Golgi, and some population of YPT1 colocalizes with late Golgi. And just recently, because we got the comments back from the referees and they asked us to use X7, that is a more widely accepted bona fide marker for late Golgi. So we did this tagging, and we saw this pancake distribution as well. And sorry, I cannot show you yet the colocalizations because this is currently undergoing, but it seems the distribution looks very similar to BPS 52. Although surprisingly, when we treated SEC7 tagged with GFP with Riquelme A, the signal went to the Spitzenkorsor. And we just got this yesterday, so I really don't have still any hypothesis of why that is happening. But I think there is some other routes going on in filamentous fungi involving YPT1 and SEC7 that will give us hopefully some clues in the future on how is traffic of vesicles to the Spitzenkorsor. We had the doubt if we could be seeing early endosomes instead of Golgi in this YPT1 labeled cisternae, so we did a co-expression with YPT2, the homologue of RAP5, an early endosomal marker, and we practically didn't see any co-localization, as you can see here, by the ICQ values of -0.5. So yeah, we're not looking at endosomes really. So it seems this is the model that we are proposing. think it's along with the cisternal maturation model of the Golgi, that some early cisternae, by acquiring new proteins, they keep advancing and maturing until they convert into late Golgi cisternae. And the big gap now is to know what's happening between late Golgi and the Spitzenkorper, or to find out what is the route going on here. And hopefully we'll work on that in the next years. And finally, we are also interested in understanding how vesicles travel. And I was mentioning before that vesicle traffic is a very fast event. And in this FRAP analysis, what we did is to FRAP the whole area of the Spitzenkorper where you can see fluorescence. So here in this panel is just after 5 seconds of frapping, of killing all the fluorescence, of burning all the fluorescence, and then after time, you keep looking at how this fluorescence is recovered. So we did that for YPT1, for FM464, for several markers we had of the Spitzenkorper.
Was protein synthesis blocked or not?
No, it was not. So basically, it's just the time that other proteins that are there are taking to
So you don't know if it's pre-existing proteins are really made?
Yeah, we don't. Mm-hmm. We're just calculating the time of replenishing, let's say, the vesicles in the Spitzenkorper. To calculate the half time, how much time do you need for the Spitzenkorper to be full of vesicles again? And we calculated, we obtained half-time recovery values that go between 10 seconds to 21 seconds in these cases. So that's quite fast. In 10 seconds, half of the content of the Spitzenkorper, well, all of this content seems to be lost and gained at the same time. So it seems to be a homeostasis of vesicles that go in and then go out. And by confocal microscopy, we really cannot measure the traffic of vesicles, or we cannot even see individual vesicles moving. But by surface microscopy, we could get some values. So in this case, you have here some big cisternae that we think are Golgi, these ones with the arrows, and these arrowheads point at some really small vesicles. And you have the zoom here, so these are the ones that we think are Golgi, and these ones here smaller, I think these vesicles. And when we calculated these with chemographs of this vesicle moving, so this is a reflection, each of these lines, on the position of one of these vesicles over time. So basically, lines that are completely vertical means that they don't move, vesicles that don't move, and the higher the slope, the larger the slope means that they move more. Okay. And we calculated some we saw some vesicles that move in an anterograde and retrograde manner from 0.4 to 4 microns per second, whereas the cisternae that we think are Golgi move only anterogradely, and they had moving values of less than 0.5 microns per second, so they move much slower than the vesicles. But again, this, remember, is self-analysis, so we're only looking at the periphery of the cell. So there's a lot of movement of vesicles going on under that layer, and we need probably some sort of real-time confocal microscopy to get all these values. And this is some turf we did some years ago of chitin synthase 1, but in this case we're not in perfect turf, because you can see here that you can see the Spitzenkorper. So you are not really in the periphery, but I'm just showing you this so that you see all this movement, constant movement of material going up and down in the cell. And for this we got values of also about 4 microns per second, as I showed you before. One of the big questions is what is the cytoskeleton that determines this movement? Are they moving by Brownian motion, or is it really a cytoskeleton involved into this? And if you look at most of the books showing hyphal cells, they all show the microtubules, which are shown here in this image. This is microtubules labeled with GFP that converge to the apex of a cell towards the Spitzenkorper region, even if in some cases they don't touch the microtubules, don't touch the Spitzenkorper. And the general accepted view is that vesicles move along microtubules until they reach the Spitzenkorper. They are dropped onto the Spitzenkorper where they are acting microfilaments, and from there they go to the tip. But nobody has really seen that. That's really nice, and it's very nice to believe it, but we really haven't seen vesicles moving along microtubules. So when we expose cells to benomyl, a microtubule depolymerizing drug, what we saw, first the Spitzenkorper disappears, but this is because the cell is bulging, it's losing the polarity, and then there is no more Spitzenkorper seen. But as soon as it recovers polarity, even if there are no microtubules seen, here it's only tubulin subunits, you can see arrival of chitin synthase 1 to the tip. So it doesn't seem they need the microtubules, really, the chitin synthase subunits, to get to the Spitzenkorper. When we use actin depolymerizing drugs instead, cytokalasin A of latrunculin A, then we saw a complete mislocalization of this chitin synthase 1. And more recently, we look at LifeAct GFP, a strain that has been obtained by a colleague of mine at CICESE, by Rosa Mourinho's lab, and you can see that this LifeAct coincides with the core of the chitin synthase in Neurospora. So we are now collaborating with her group to look at the role of myosin and actin in transport of chitin synthesis. And when we look at myosin 5 also localizes at the Spitzenkorper, and we did a construct in which we eliminated this DIL domain, which is the cargo binding domain to the vesicle, and we substituted it by GFP, so that in theory the vesicle can no longer transported by the myosin 5. And what we saw is that myo 5 still reaches the tip, but the phenotype is severely affected. So it seems that at least secretion or transport of vesicles that are important for growth is severely affected. Even the myosin can arrive with no cargo to the tip. And we are now analyzing this further. When we express chitin synthase 1 into this sort of mutant strain, this strain with no cargo binding domain, because it was a knock-in male strain, so there is no other myosin 5 in the cell, we saw that chitin synthase 1 could reach the tip, nevertheless. So at least chitin synthase 1 doesn't depend on this myo 5 for transport to the tip. So I'm going to summarize what I've shown you. You've seen that the differently sized vesicles in the Spitzenkorper contain distinct cell wall synthesizing enzymes, and they have a layered distribution. Cytosomes with chitin synthase are in the core of the Spitzenkorper, and the macrovesicles surround this core. We've seen also that an exosome-mediated mechanism is needed for the last steps of exocytosis to dock these vesicles of the Spitzenkorper. We think macrovesicles, but we still have to prove that they are not microvesicles, to this apical plasma membrane. And lately, we've seen that RAB-GTPases participate at late secretory events of this ordered delivery of the vesicles to the Spitzenkorper. YPP1 associates with microvesicles at the core of this Spitzenkorper, while YPP31 and SET4 associate with these macrovesicles at the outer layer. And finally, we've seen that this RAB-GTPase localizes also at early and late Golgi cisternin, which indicates that Golgi cisternal maturation model, of course. So we're starting to understand a little bit what's going on in the cells of Neurospora, although this is a diagram made with information got from other fungal species. We still have a lot to understand. We're just starting to understand what the vesicles of the Spitzenkorper contain, but we're still far from knowing how they reach the Spitzenkorper. We're working on that, as I've shown you. And what we don't know is why do they remain there at the Spitzenkorper? Why don't they immediately reach the plasma membrane and get exocytosed? It must be a mechanism to regulate that there is not too much getting at once at the tip to maintain this nicely regulated growth. And we're also starting to understand how they reach the plasma membrane and where vesicles are generated. So I want to thank the people that have participated in this work. The work of chitin synthase has been done by two of my PhD students, Rosa Fajardo and Eddie Sanchez, and Eddie also has done all the work on the RAP GTPase. My colleagues at CICESE, Salomon Barnicki and Rosa Mourinho Perez. And then I have a long list of collaborators around the world which are having key to really be able to complete this project. Robert Robertson at Arizona State University is the one that has done all the electron microscopy work. Michael Freitag and Erin, his student at Oregon State, have helped with the CICESE work. Peter Novick, I did my sabbatical in his lab about a year ago, and we work also in the CICESE proteins. And well, I have a long-standing collaboration with some German collaborators on a project that has been funded by the DRG from Germany and CONACYT from Mexico. And finally, I want to thank the Fungal Genetics Stock Center for Strains and the Neurospora Genome Project for some of the knockout mutants. So thank you for your attention, and I'll be happy to answer any questions.
Thank you very much for this beautiful and colorful talk, Meritxell. We have pretty good intense with it. Questions?
So you mentioned that we don't know why the Spitzenkorper, why vesicles kind of paused there before going on. Would the exorcist play a role in that pause?
I think it's all related, right? But I don't know if it's the signal that really triggers that they pause there. I think there is something beyond that. I mean, of course, we have only looked at this deletion mutant, and then you see that vesicles don't even reach there, right? They don't accumulate because you're really interrupting a very important step. But in terms of the exorcist regulating that pausing, I don't know.
So you're not seeing the accumulation of the Spitzenkorper in the exorcist. So could they just be going to the cell surface and fusing then instead of accumulating in the Spitzenkorper? So have you looked at like chitin levels to see
on
if
the exorcist mutant?
Yeah, see if chitin is reduced.
Yeah,
maybe if chitin is still being made at a well type level, maybe you're just not getting pausing at the.
Right. No, we haven't looked at that. We can look at that. Yeah. I don't know who was first.
With regard to the dichotomy of delivery to the hyphal tip versus going to the site of septation, at least in other systems, that's often directed by a foramen, which is needed to polymerize the actin filaments on which any myosin 5-site motor would run. So if you looked at where, say, BNI1 and BNR1,
so I haven't, but
localized.
I haven't, but other labs have. And if I remember right, they only find it at the tips, BNI1. Do you know? Do you remember? No. I think they only find it at the tip. But a lot of the other proteins that are involved, other myosins, because of course, in the septum there is also the contractor ring. So a lot of the proteins that regulate that are both at tips and septum. But in case, for example, polarisome is only, some components are only at the tip. So
I would be surprised if there isn't a BNR one.
BNR?
Yeah.
BNR, I don't think there is a homologue in Neurospora. Has it?
In that same regard, like Tony Brecher has shown that for vesicle recognition by myo5, it needs to be properly coded with PI4P. So one thing that could be limiting in the Spitzenkorper collection of vesicles is the rate of synthesis of PI4P. Had a chance to look at that?
I haven't. Yeah, they have for other fungi, for Candida, I know that they have, and I don't know if in Aspergillus, I think they also have started that, but now we haven't looked at it. That's very interesting.
And one last, you were able to nicely fractionate on some gradient, I forgot what it was.
Yeah, sucrose gradient.
Chitin containing from the glucan synthase containing. If you take those fractions and use your GFP tag as a handle to pull down those vesicles, what do they actually look like in the EM number 1? And if you do mass spec on those, which yeast snares are present in those?
This is my next 5-year project. No, we haven't done that. Well, we did one, I think we did a run-through of one fraction just to see. but we haven't done, replicate, but we want to do that, to do the fractionation and then look at both the proteomic content, but also the lipidic content and maybe the RNA content.
Yeah, is it known if excitosis is controlled by circadian rhythm? And do you for experiments in complete light or is it cycle?
Probably they are, but I had very close colleagues doing those analyses, and I'm not up for 24 hours in the microscope looking at the up and down levels. But I'm sure they are, because during the cycles, right, during Neurospora circadian rhythm, there is a time where the conidiation is increased and then the apical growth is reduced because all these conidiophores are formed. So I'm sure that the levels of expression of certain proteins change during those times, during the banding process that we see in Neurospora. But yeah, no, we have not done that.
The macrodescal movement separation of microdescal, well, there must be chaperones, there must be the partners want to do the
Yeah.
Do you know anything about that, or have you been able to find these where they're one has been localized, or what are the signals for
So we looked, based on also work from Trevor's and Randy's lab, we looked at the sequence of all the chitin synthase, but we don't find the same domains that they have reported in Saccharomyces for the recognition with either this exomer code or the other adaptor protein code. We haven't done that for glucan synthase because up to now, just within the last year, we got finally the catalytic subunit expressed. But yeah, I'm sure that there must be something encoded in the sequence, the amino acid sequence, that determines that they go through recognition of one donor or not. But yeah, we haven't done that yet. And as you know, we still don't have any TS mutant nice to be able to switch on and off the expression. But we look at some of the deletion mutants from the library just to see if we could get interruption of some of the secretory pathway step, but so far we haven't. I think we have one that is that is a homolog of the chitin synthase 3 in this, one of the CHAP proteins in this, and I think we have expressed one of the chitin synthesis in this background mutant, and then it cannot exit the ER. So that's the only thing we have right now that looks promising.
Yeah, so what I found pretty intriguing is that SEC7, there was a GP, suddenly appears as a when you at DFA, so why is that? Do you have any hypothesis?
No idea. I got that slide yesterday.
Okay.
And I just show it. I don't know if anybody has some suggestions. But I really...
Is there a chance that that SEC7 sort of travels through this pathway super quickly, and then that some sort of gap activity is also
so you are impeding, because it has an effect on the Golgi-associated SEC7.
Right.
But then, since you're not letting it, since you're blocking all the Golgi, maybe it's just using an alternate pathway where early endosome recycling is taking place. I'm assuming it's linked with endosomes, but...
Yeah, that's just, if the cell is really directing that, or is it just weird side effect?
Yeah. Because when we use Briffeldin on the chitin synthase tax strains, we actually see more labeling also at the Spitzenkorper.
Okay.
The SEC7 at the core or the ring?
I think the core. Mm-hmm. Yeah.
Good. More questions? If there's no more questions, I think Meritxell, one more time.
Thank you.